Welcome to Data and Biotech, a podcast from Cordyne where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Here we go.
Welcome back to Data and Biotech. I'm Ross Katz, data science lead at Cordyne. There's a story the AI drug discovery field likes to tell about itself, where generative models search vast chemical space, identify novel compounds, hand medicinal chemists short lists of candidates ready for synthesis and testing. It's a compelling story, and in some respects, it's true. But spend time with a computational chemist who's been doing this since 1985, who's worked at what became GSK and Pfizer, who's shepherded a compound all the way to a marketed drug, and a more complicated picture emerges. Because the challenge of finding a molecule that binds to a target is only one part of the problem. The molecule also has to be metabolically stable, non-toxic, orally bioavailable, and maybe most importantly, it has to be makeable. And that last constraint, synthesizability, turns out to be far harder and far easier to underestimate than most people outside the lab realize. My guest today is Paul Finn, Chief Scientific Officer at Oxford Drug Design, a computational drug discovery company with roots in Oxford's chemistry department and a decades-long commitment to making sure that what the computer recommends can be made in the lab. Paul brings a rare combination: deep technical fluency across classical and modern computational methods, and a practiced skepticism about AI results that look too good to be true, because he's done the work of checking whether they are. Today, we'll get into what it means to search chemical space responsibly, why chemistry has proven so much harder for AI than biology, and what a rigorous synthesizability constraint approach to multi-parameter optimization looks like in practice. Paul, welcome to Data and Biotech.
Thank you, Ross. Very pleased to be here.
Awesome. Well, to get us started, you started your career at what's now GSK in 1985, and you moved to Pfizer, and then transitioned through a series of Oxford area spinouts before landing at Oxford Drug Design. So you've got this 40-year arc from classical medicinal chemistry to modern computational methods. I'm interested in just what that journey looked like, and what drew you toward the computational side of discovery.
Yeah, so I started off as a biochemist, doing a classical biochemistry degree, but I was always more interested in, if you like, the theoretical aspects than the practical ones. Never so great in the lab, and that ultimately led me to my PhD research topic, which was in the prediction of protein structure from sequence. And this was back in the early 80s when computational approaches were still very much in their infancy. And as a side project to that, I got involved in a little sort of structure activity study predicting the activity of peptide hormones, and so that got me interested in drug discovery, and so I went straight into industry to apply those methods, and I've been applying them to drug discovery ever since, through those companies that you mentioned. And currently at Oxford Drug Design, that really just continues. We have a strong activity in developing novel computational methods, combining the new Gen AI ones with traditional ones, and then applying them to our internal pipeline of projects which are focused in on oncology and antibacterials.
Yeah. So can you give me some examples of like where some of the methods that have been around historically are like allow you to see the emergence of generative AI and the computational methods that are more generative in nature through a different light, and or like understand sort of the gaps that those methods might expose?
I think that they all really fall into to characterize that in a straightforward way, really making checking, providing checks of what is being produced by these generative AI methods, which still are to a large extent black box type methods, it's really difficult to get a firm handle on why these particular molecules are being suggested by the method. So if you can then apply some really rather straightforward methods in terms of just generating molecular conformations, or just calculating simple properties from those methods, and then say, well, does this make sense? Is this, could this be could we find plausible reasons for why these molecules have been suggested, and that helps give a lot of confidence that they're not just some hallucination of the particular AI method.
Yeah, and I mean, one of the things that you mentioned previously as well was just this idea that it's great that generative AI can produce a small molecule that in principle does what you need it to do, but there's also the synthesizability problem that is sort of underestimated. So, you've got generative models that are searching chemical space, but chemical space is drawn on paper, and chemical space is something a medicinal chemist can make, are very different things. So I'm wondering, what have you seen happen when synthesizability is sort of treated as an afterthought, and then on the other hand, what changes when you have to think about it as a hard constraint?
Yeah. So the chemical spaces that can be searched these days with the generative AI methods are extremely large. But there are a couple of aspects that really make them difficult to apply really effectively, and that's because, exactly as you say, at some point, a medicinal chemist or a synthetic organic chemist is going to have to make that structure so that it can be tested to verify that it's really got that activity. And I think it's often underappreciated how not straightforward it is to just be presented with a molecular structure and then being told to make that. The link between the chemical structure and how easy it is to make its synthesizability is very complex, and just changing a single atom in a structure can change it from being very simple to make to being very, very difficult to make. And so what often happens is that all of the work that's gone into that generative AI and the extremely large amounts of computation lead to molecules that cannot reasonably be made to validate those methodologies. So if you consider the synthesizability from the start, if you somehow constrain that search so that it will only make molecules that suggest molecules that can be made, this is a much more effective method and much more practical method to move forward to test the ideas. And I think this is also related to exactly how the AI methods capture that chemical structure in the first place. I think that also causes the link between the chemical structures and their biological activity to really be quite difficult to fully model in the generative AI approach.
Can you unpack a little bit about why that is, and then I want to return to like this idea of synthesizability, like why, what is it about the way that generative AI models tend to model
Sure.
chemical space that lead to that problem?
Sure. So when generative AI was first being applied to chemistry, and still to a very large extent, the key question is, or the first question is, how do you represent the chemical information, the chemical structure information, to the generative AI model, whatsoever that model is? And because so many of these the deep learning methods have been built on things like large language models, which are dealing with text and strings of characters. A lot of the early work was done, and much of the work still being done is representing the chemical structures in that symbolic way. So effectively, for the chemical structure diagrams that you see in textbooks and in publications, effectively it's a two-dimensional representation of the structure. And the problem is that that's a highly abstract representation relative to what actually happens, the three-dimensional chemical structures that are really interacting with the protein target. So you have quite an abstract chemical representation, and then the AI methods themselves are generating these very abstractions of that into these spaces to generate their models. So the link then between the chemical structures and what really matters in terms of their activity, a real three-dimensional molecule interacting with a real three-dimensional protein, that makes it very hard for the AI to capture what's really important, and there, as a result, you get predictions that very frequently don't pan out when you test them in the real world. And so constraining the chemical space, I think, really helps, and representing the chemical space in a way that is more closely related to the physical reality, is really what's helpful. And there's some movement in that direction, but it's still not the predominant way in which this is done, I would say.
So starting with constraining the chemical space, my understanding of this is that you kind of have to have a sense of what the different building blocks are that you can synthesize, that you can put together, that and knowledge of the methods that are out there. And it's like sort of a combinatorial problem of figuring out like all of the different molecules that hypothetically could be made, and then fitting that into the specific problem that you're trying to solve in chemical space. But that one of the challenges here is that when you constrain the chemical space, then almost by definition, it's much harder to find some novel approach to solving a particular binding problem or a particular therapeutic problem. So I'm just interested in how do you think about first of all, am I understanding sort of the nature of the problem correctly? And then, if so, how do you navigate the trade-offs between like ensuring the synthesizability and making sure that you're able to find something novel that's able to take a different approach that can work?
The constraining of the chemical space involves synthetic reaction schemes that join building blocks together like pieces of Lego to make more complex structures. So it involves having a well-defined set of chemical reaction types that are well understood and precedented and work. It requires having a list of all the potential reagents or building blocks that can go into those schemes, and then working out what could be made in this potential virtual space. You're quite right. It's a valid criticism that taking that approach does shrink the size of the chemical space from the entirely abstract one of all theoretically available molecules. However, I think it's really a theoretical rather than a real limitation because of the combinatorial nature of the chemistry and the way that you can combinatorially combine building blocks in a sequence of reactions. Those chemical spaces are still extremely large. 99.999% of them have still never been made by anyone ever in the history of mankind or humankind. So you're still searching extremely large spaces, and there's plenty of scope to find novel chemotypes that have not been found before. So, and I think the other practical thing, I mean, if you look at what chemical reactions medicinal chemists use, there was, I think, a few years ago now, a paper which just reviewed all the patent literature and said, well, what chemical reactions, how do chemists actually make these molecules? There are about 10 reactions that dominate the entire thing. So, and that's because those reactions work reliably, and there are a lot of reagents available to do them. So I think that this is a very viable approach, which still leads to very large chemical spaces, and gives you a good opportunity to find novel, patentable chemical matter from them.
Yeah, that makes a lot of sense, and obviously, there's there's a balance between over constraining yourself and under constraining yourself, but oftentimes when you select the right degree of constraints, you're able to find the correct combination of like a space to be creative, and then also, you know, knowledge that you're going to be able to get the characteristics of the molecule out the other end that you actually want from the
Exactly right.
process.
And maybe just one other comment, because you know, yes. So, and indeed, you know, obviously you can expand the chemical space by just increasing the size of those, you know, the number of reaction schemes you have, and the reagents, and so I think you know, building more sophisticated, more sophistication into those schemes, because again, another you know, underappreciated point, I think, is that even when the synthetic schemes are well known, they're published in the literature, and people have obviously used them to make molecules. You go into you try to reproduce that, or you try to use that scheme to make a slightly different molecule, and you find the first time you tried it, it doesn't work. You know, so you have to tweak the conditions a bit, or you have to change the solvent, and you get it to work. So these things are really, you know, the synthesis is really very, very challenging. And you know, I think one of the areas we're working on is really trying to build those sort of synthetic spaces to try to make them more and more sophisticated, to build in some of these aspects, so you can expand the size of the database, but also improve the reliability that you will actually be able to make them at the end.
Yeah, I I'll just ask now, you know, what are some of the ways that you can think about both improving the size of the database and improving the reliability?
So I think the size is the easiest part. I mean, there are, you know, medicinal chemistry is very active. You know, many new, you know, synthetic methods are being discovered. Uh, Many of those will be amenable, you know, to generating molecules that could be of potential interest. So one is, you know, just the task of keeping track with interesting reactions that come up in the literature. The other one, which is much more complex and in some ways more interesting, is how to encode the reactions in a more sophisticated way, so that, for example, you know, this reaction works with reagent A, it doesn't work with reagent B, even though reagent B seems to have the right chemistry to do that. That's because reagent B has some other chemical group which acts as a competing for that, and leads to an impurity being generated. So making the rules more sophisticated for generating the library, so that you have fewer of these examples where actually, you know, it doesn't work at all, or it does work but you produce a large level of impurity, which just makes it not practical. So
Yeah.
one of those is just accretive, and the other is actually improving the technology to improve the quality of the virtual database.
Yeah, no, I'm just imagining that it is a hard problem to keep track of all of the different new discoveries that are being made in terms of it, and also to apply sufficient rigor to the validation of the results before you're incorporating them into into the library, because there's this entire spectrum of results that would either more effectively constrain the space or would expand the possible chemical space, that, you know, you sort of have to weigh the evidence with regard to incorporating those changes into this. I'm imagining a very large rules engine for how the combinatorial problem ends up being computed. Am I thinking about that right?
Yes, yeah, absolutely. And again, you know, everything we do is sort of driven by pragmatism, really. So, you know, I guess if a reaction sort of seems to, you know, come up several times and so on, and this looks like, and again, another point is just how many reagents there are. You know, if you get a lovely reaction, but if, you know, there are only 3 of group A and 3 of group B, then, you know, adding 9 compounds to your virtual database is probably not going to be worthwhile doing. So it's a whole bunch of pragmatic and practical things. And then if that reaction, you know, seems to be generating interesting molecules, of course, then you can go on and further refine the definition to improve the quality.
Yeah, no.
When you know that that's a reaction that actually might be useful in the future.
Yeah, that makes a lot of sense. I want to turn back to this idea of understanding and modeling um, molecules and their reactions better in 3D than generative AI models typically model them with sort of the language-based approach. I'm wondering if you can sort of introduce us to some of the ways that we can think about how you can model in 3D that more effectively capture how the structure of the molecule is going to relate to its its activity and its interaction pattern.
Yeah. So I mean, in some senses, that's very straightforward because it just builds on all those historical computational chemistry techniques that we were talking about at the beginning. So generating a three-dimensional structure of the molecule where each of the atoms is in a physical three-dimensional space, and the atoms are joined together by their bonds. So you have a three-dimensional object. I mean, those techniques have been well established for a long time. So representing the structure to the AI as a three-dimensional object is in some ways straightforward. However, there are still a lot of complexities to that. It's not so shape. The shape of the molecule is probably the preeminent sort of property that drives molecular recognition and binding. You have a shape of the drug, you have a shape of the cavity into which it's fitting, and they go together in a complementary surface. And the more complementary they are, the strong, other things being equal, the stronger the binding, and so on and so forth, which is a good thing. But it's not just the state, the shape. These molecules have got a electrically charged, and they've got electrostatic potential around them, which also is involved in the strength and orientation of the interaction. And then you have the additional complexity that most molecules of pharmaceutical interest are not rigid bodies, that they are able to change their shape by rotating about the bonds and adopting other shapes. So you actually have that for any individual molecule, you have a plurality of shapes that they could adopt. So building all of those and representing those to the generative AI method is at the first level simple, but then as you try to make a more sophisticated model, it becomes more complex. And especially that's true if you add the protein side to the equation as well, and are trying to represent the shape of the cavity into which the molecule fits. But at the end of the day, that is much closer to what actually happens, the real physics of the interaction, and therefore, despite those complexities, it seems to, in our view at any rate, that is a better chemistry representation to be using than these abstract, linear, string-based representations, which are very far divorced from that physical reality.
Well, I think I came across that some of your colleagues at Oxford are using like topological methods to sort of model the shape in different and maybe more nuanced ways than we were modeling it previously. So I'm just interested in understanding from your perspective, like, where is our understanding of these different elements sort of limited, and where is research trying to push in terms of exposing more and better data information to the models in order to understand that binding behavior better?
Yeah. So, I mean, I think in principle, the physics of what's going on when a molecule binds to a protein are pretty well known. Yeah, they're pretty well worked out. But applying them, is in sufficient rigor is what's difficult. So in the limit, the quantum mechanical description, is going to be the most accurate one, but it's largely impractical to apply just even in the current state of computational power. So we use approximate methods, but they're still pretty accurate. But what's difficult is that there are yet other factors which we haven't yet discussed. We've discussed a little bit the fact that molecules are flexible and the proteins are flexible, and that creates lots of additional complications. The other complication that we've not discussed yet is that these reactions or these interactions occur in an aqueous environment. They occur in water in the cell, but it's not just water, it's stuff with lots of counter ions and other proteins and so on and so forth. So the role of the solvent in this in this interaction, how that is involved in stabilizing or destabilizing the interaction between the drug and its protein is also very, very complex. So I think we're still some way from being able to, at an atomic level, simulate that well. However, but work is going on in that direction. In the other direction, trying to make simplified models that are actually tractable. Yep. So, for example, calculating a couple of things just taken from what we've been doing. Other people are doing similar things. Is to generate from those 3D molecular shapes a compact representation of, so rather than having to keep the 3D coordinates and all of that electric charge information, try to generate from that something which is a more compact summary of that information that is more amenable to database searching and so on, but still contains the essence of the interaction, albeit with some noise introduced. And the mathematical.
Should I think of that as like a graph-based embedding, or is there some other way of compacting that you're talking about?
Yeah, I mean, you can certainly do that with graphs. We do it in a we're doing it in a slightly different way in generating the vectors of numbers that are chosen to reproduce the shape and electrostatics at a particular level of resolution. And I guess there's not time to go into the details of how that works, but
sure,
it's very, very useful to generate these compact, simpler representations, because the molecular comparison then becomes much faster and the data storage becomes much smaller. Just very briefly, the topological work, I think, is also directed at that, but also in some ways it's looking at topological invariance at different scales. So in what ways do the shape or properties that you can calculate from the shape stay the same or change at different levels of spatial resolution? And that is in principle a way in which you could maybe start to encapsulate some of the things which the molecule keeps if it changes its conformation, for example. And that's another way of simplifying the structure, but it's like an orthogonal way to simplifying it, so that you're getting two simplified representations, but they're capturing different aspects of the 3D structure.
Yeah, that makes a lot of sense, and it sort of aligns with what I'm hearing from across the model-based conversations that we're having about, it's all the different lenses that you can take on a given phenomenon that are orthogonal and that allow you, and then you just have a compact representation, and then you concatenate those compact representations together, and that hopefully feeds more information into the entire modeling system. If it's okay with you, I'd like to switch gears a little bit. So you're searching chemical space for molecules that have certain characteristics, certain potency, metabolic stability, solubility, selectivity, toxicity, and we mentioned synthesizability. I'm interested in how many properties you're trying to optimize simultaneously, and then how do you approach that problem of optimizing across all of these different parameters simultaneously?
Yeah. So yes, that's right. I mean, drug discovery is always any individual project is always a multi-parameter optimization. And the clinical candidate is never the one that's the most potent or the most soluble or the most stable. You're always searching for this compromise of properties that are where it's good enough against all of the criteria that need to be considered. So, and it's very that list is very long, so it's actually very hard to try to do this in a comprehensive way, and I actually think rather foolhardy to try to put all of the things that you want into the calculation. I think there are just too many, and we don't know enough in detail about some of those that the models could be any good anyway. So again, we focus on the things which are for the particular project we think the most important, and focus on those. They can often be pulling in different directions. So, making the drug more hydrophobic will probably improve its potency, but it will also probably make it less soluble, and you need both of those. So you have to deal with those, the fact that they're pulling in different directions. Again, I think it's by tailoring the scoring function to your particular problem. You can weight the different parameters to some degree, so that you might be more keen on maintaining potency than on solubility, or maybe your solubility is already good enough, so you don't need to quite weight that so much, and that way that you can make them maximize the potential of those, for optimizing those properties. And again, the other important thing, of course, is to check back frequently with experiment to make sure that you're on the right track, in this optimization, because it's quite easy to go down blind alleys if you don't do that.
Yeah, I mean, in terms of sort of the problem specific nature of choosing which aspects you focus on, I think it might be useful at this point to ground in, the specific therapeutic areas and approaches that Oxford Drug Design is doing right now. So, my understanding is you're working in two therapeutic areas. There's new antibiotics to address antimicrobial resistance and oncology, and that, if I understand correctly, it's you're using sort of the same molecular function. So I'm wondering if you can just talk, or similar molecular functions. I'm wondering if you could just talk to us about these programs and these areas, and then maybe we can return to sort of the nature of the problems that you focus here, and like how you think about optimizing for these different therapeutic areas.
Yeah. Yes, that's right. Those are our two therapeutic areas of interest. I mean, in some ways that they have some similarities. They're both about ultimately, killing or stopping the growth of particular cells, but in one case they're aberrantly proliferating human cells, and in the other case they're undesirably proliferating bacterial cells at some infection site, whatever. So there are some levels of commonalities, but of course the individual approaches are quite different. The targets, even if they're similar, are different in those two organisms, and so you have to deal with that. I guess in terms of the, I guess actually probably a useful one is, if your target, as they often usually are, is inside your protein target is inside the cell, the molecule needs to get inside the cell to reach that target, and so actually that occurrence, getting sufficient concentration of the drug inside the cell for long enough to have the desired effect, is quite a challenge, particularly, I guess, in the bacterial programs, because bacterial cells are very different from human cells. Their structures vary from organism to organism in ways we don't have time to discuss, but essentially they have a complex cell envelope or wall around them that is a very significant barrier for small molecule drugs, which is what we deal with, getting across that barrier into the site of action. And discovering new antibacterials has proven to be extremely difficult. Not many new antibacterials have been approved in recent years, and almost always this lack of accumulation into the cell is what is the failure. It's very rarely failure to find a drug that a compound that doesn't bind strongly to the target. It's overcoming this formidable cell wall barrier that's the main issue there. And there are other similar issues, but to much lesser extent. So that's a clear differentiation in terms of the design approach with these two projects. We've had to think much more about this issue of cellular penetration in the antibacterial projects than we have in the oncology projects.
So how does that enter like the optimization problem? Are you sort of actually? I think a good way of talking about this would just be sort of orienting us to the drug discovery, drug design and discovery process that you go through, and sort of like how that cellular penetration element enters the process for selecting the next set of candidates that you're going to synthesize, et cetera.
Yeah. So the way this works is that you have what's usually called a screening cascade. So you have a series of tests, a series of assays that you run on new compounds, and then if they have a good result in those assays, you take them on to more complex assays and so on, and usually more expensive assays and so on, to work out how close they are to becoming a clinical candidate. So what changes is effectively the assay pipeline. In the antibacterial case, you have assays that are giving you a readout with regard to the cell penetration. Yep. So that can be done in a simple way. So, for example, you have a compound that's a very, very, very potent inhibitor of your protein target, but when you administer that to bacteria, it has absolutely no effect. Yep. So plausibly, the drug is not accumulating in the target. So that gives you some information, and so you could try to change the structure in a way that it might accumulate. But you can then develop more sophisticated assays where you take, for example, mutants of the bacteria where some aspect of its cell wall is mutated, so in a way that makes it easier, for example, for compounds to get in, or makes it harder for the bacteria to pump them out, or in various ways. So now you've got a more sophisticated screening cascade, which is giving you more information about, well, what actually is the problem? Can it not get in? Is it metabolized when it's inside? Is the bacteria pumping it out because it's very good at these efflux mechanisms? And then you can start to build that data into your model. Yeah. So you're and indeed mostly you're not optimizing for activity against the target very much at that point. You're just maintaining that at some reasonable threshold, and it's really these other properties that are going into the optimization.
Yeah. So as I understand it, you're using Bayesian optimization. You're sort of like, this is an active learning loop where you're choosing sort of the top set of candidates to go and run your assay cascade against, and then feeding the data back into the model.
Yeah. So I explain how that works in outlines. So we've got this synthetic accessibility based approach. Yes. So we have. Yes. Reaction schemes, we have reagents, we have a chemical space that we can build, and so as we generate data, we start to be able to associate that data with particular structures in that virtual space. Yeah,
right.
And so then the way we approach it, in contrast to the classical way where you've got this enormous space and you're just picking molecules out of that space that are in some ways already defined, what we're doing is we're searching, if you like, the reagent space. What combination of reagents is associated with the best activity? Yeah. So that's really what the Bayesian optimization is doing, to focus on those patterns of reagents that lead to the molecules that are associated with high levels of activity. Yeah. So there's a Gaussian optimization process, and Gaussian process model and the Bayesian optimization help as we build the data to focus on those regions of chemical space where the activity seems to lie, and those then generate the particular examples of the virtual library which we make, and then of course we test those, and then we get more data into the model and iterate in that way.
Yeah, and my understanding is that one of the benefits of the Gaussian process modeling is that it gives you sort of a measure of uncertainty that you can use as part of navigating the trade-offs between exploration and exploitation of the knowledge that you've gained. Can you give us a sense of of how uncertainty is measured inside of the process that you just described?
Uh, oh, it's actually quite tricky to do in a sentence or two. Um, but I, I guess we're looking at the sort of nearby reagents and how likely it is that this would would lead to an activity that's within a certain level of what we already know. It's actually quite hard to quantify because of the discrete nature of what we can make. But I think I think we get a pretty good idea if the signal is pretty flat and not very strong. It's certainly not at the level where you have very high degree of confidence in any of these predictions. There's certainly lots of false predictions, but it's good enough to make progress. And I guess at this current stage of development of these methods, I guess that's all we can hope for at the moment. It can, and we have found interesting improvements in the structure through these sorts of approaches. And there's many ways which we are in which they can be approved, and we're trying to approve them. But
that makes a lot of sense. I think for Gaussian process modeling has been around for a very long time, and deep learning is all of the rage right now. And I know that you had a 2023 paper in the Journal of Molecular Modeling with Ravneet Jagdev and Thomas Madsen, where you took the Stokes et al. Hallison paper apart in a specific way, and you showed that Hallison can be recovered from the same data set using methods that are a lot older than deep learning. And so, I'm interested in that result there, and what conclusion you come to about where you want sort of these data hungry methods like deep learning in a drug discovery process versus where these sort of tried and true methods that are older and maybe sort of more principles based are the right approach.
Yeah. I think it really comes down to validation of the methods. So the many of these generative AI or deep learning methods, are very complex. They're using a lot of computer power, and they're generating models with very large numbers of tunable parameters and so on, and they can produce results that superficially look extremely good. Publication on Hallison is one example of that, but there are many others where with completely different approaches to deep learning or generative AI, where the publication seems to indicate that what the method has actually learned. Going back to what we were talking about before in terms of binding, the method does seem to have learned the physics of the interreaction of the reaction between the ligand and the protein, because in all their validation they got very high correlations with the data sets that they tested. However, subsequently it's proven that those results were artifactual and were arising from something else. And those something else's are frequently in the preparation of the data. There are many ways in which these sorts of the chemistry data sets can be biased, and that just improves the chances of getting a false correlation, for example. So these things are very difficult to do, and I think looking at and the data generation of a good quality data set is difficult to do. But also, one thing which they didn't do in the Hallison paper, and I think is frequently lacking, is benchmarking it against a really simple methodology. There are lots of simple methodologies that could be applied in most cases, and it's really the improvement you get over those simple methods that are telling you to what extent all of that extra complexity and sophistication and computer power are really doing anything useful for you.
Yeah, there's a couple things that you said there that I think are really valuable to just press on a little bit. Obviously, like having a valid baseline comparison for the method that you're using tells you how much of it is the complexity of sort of the mechanism that you have to pick up in the data in order to get the information out. And that's what we're used to happening with deep learning, is that it's able to, by nature of having so many parameters, pick up on patterns that simpler methods are just not able to pick up on. And how much of it is also just is it overfitting to elements that have leaked between your training and your test set in the training of the model. And so there's just a degree of care with the data set and with the methods that's needed when in chemical space that seems like it's sort of a lost art form, with sort of the emergence of deep learning as being the best approach or the leading approach across so many other parts of the modeling space.
Yeah, and I mean to be fair, it's often indeed not just that it hasn't been done; it just is very difficult. So, for example, in some of these protein ligand prediction methodologies, trying to develop a method that will generally be able to predict binding affinity accurately. The data sets there from past crystal structures and so on. It's just so easy for a structure that is not you never want structures that are in your training set in your test set. But right. It's very, very easy, given all the molecular biology, for there to be a very similar structure in your test set to one in your training set, and so you're just giving the method an easy win there. So testing it on a genuinely novel, set of proteins in this particular example, which have genuinely not been seen, by the training method, takes an awful lot of work, and people have tried to do that well, and then subsequently people have said, but you didn't think about this, and oh yeah, we didn't think about that. Yeah, so it's also generally hard to do, as well as people not giving it enough attention.
Yeah, it's hard to know the comprehensive set of questions you have to ask about a model in order to be sure that it's not memorizing some artifact or hacking your reward function or doing all of the things that models are likely to do when they're consuming a lot of data and heavily optimized toward an objective. To that point, like the example within drug discovery and development is the success of AlphaFold, which was built on decades of accumulated structural data with sequence to structure relationships across hundreds of thousands of proteins. But as you gestured at earlier, chemistry doesn't really have an equivalent. So there's the relation. And my understanding is that that's because the relationship between chemical structure and biological activity is much more complex, but also that the data is more fragmented and much of it is proprietary. So I'm interested in your perspective on how data scarcity shapes what's possible computationally in small molecule drug discovery.
Yeah, I think both of those things are true. In biology, some of these sort of large language model type methods, dealing with strings of characters, work quite well for bioinformatics and so on. So I think there's a more natural match between there and the building block reservoir of proteins is only 20 amino acids and so on, and the structures consist of lots of regular repeating helices and sheets and so on. So whereas chemistry, it's far it's far more diverse in terms of the chemotypes and the sizes. It's just a lot harder problem. And in terms of the data, again, we just don't have that data where there's associated lots of chemical data, but not so much where there's associated relevant biological data. And I just reminded that was, I think it was a few years ago now, so it's maybe 3 years old, but but I think it's still true. There was a Nature editorial, the title of which was, "For chemists, the AI revolution has still to happen." And it was just, again, making this point that there just isn't that set of data. Not even for something straightforward like binding affinity. And then if you go to metabolism, where there are hundreds of metabolizing enzymes, or if you go to other things, how we could build models that could be effective in those areas when the data is so scarce and so diverse. Different labs produce different results, and so on and so forth. Quite a lot of it's locked away in big pharma vaults. So they perhaps internally, some of the big pharma companies have enough data to do something useful with some of those models, but not an awful lot. Although there are efforts to put these things into the public domain in a pre-competitive sort of way. That's still pretty much lacking. So it's an enormous impediment, I think, to generating good models in chemistry.
If you were in the position of advising a pharma company evaluating an AI drug discovery vendor, or someone claiming that their platform that can accelerate hit identification or improve synthesizability or reduce attrition, what are the kinds of questions you would tell them to ask?
I mean, I think in general, it has to be around, supporting how well, the general answer is how well have they supported their claim. So, being able to quote very, very large numbers, I don't think matters at all. But if they're saying that they can find, good structures out of these databases, then it really comes down to, prove to me that you can actually do this in the real world. So ideally, they would have done this themselves, or they've done this with an academic partner or something. There's a publication where they have shown that they have identified a molecule that wasn't known about before, or wasn't close to a molecule that was known about before, that has the activity that they were trying to find at the start. So those sorts of validations, and I think the other thing is to really try to understand what was the AI's contribution to that finding, because many of the AI papers that have been published claiming or reporting molecules that were discovered through AI, the AI component, when you read the paper, is step one, and there were 99 other steps that happened afterwards, all of which required human input to ultimately decide that was the molecule that was the one. So right. This is an area where I think the publications are really quite, not deliberately poor, but I think it is very, very difficult often to really understand what was the real contribution of that method to this, and what was the contribution of all the things that were done afterwards, and indeed maybe before, in terms of the data selection and so on that led to that. And that, I think, is often a difficult question for the companies, because it's very hard for them to, I think it's often hard for them to demonstrate that, really that the AI was the key or the major contributor to that finding.
Yeah, that makes a lot of sense. So as we head toward the end, are there any methodological unlocks in, things like representations of molecular flexibility, or the integration of physics-based constraints with learned models, or in dataset quality and curation that you think are likely to help advance AI's contribution to small molecule drug discovery?
I mean, I think there are, well, certainly we've talked about data preparation and how difficult that is. I mean, I think that's an area that still needs work. I think there are academics working on this. Charlotte Dean's group in Oxford, who are near to us, are doing some really good work on that in terms of trying to generate better data sets in their area of interest, which is in sort of protein ligand binding. I think there's a lot more that can be done there to really make these make proper tests for these methods. I think there are some areas in the chemistry representation that are still not very well treated at the moment. Those are two examples of that would be around the charge state of the molecule. Is it protonated? Is it not protonated? Is it a salt? Et cetera, et cetera. And also, technically, chemical structures can have these states called tautomers, where the atoms move around within the structure, but what's in the solution is sort of like a mixture of two different things in rapid equilibrium with one another. And in the chemistry databases, you usually have to pick one or the other as the way you're going to represent the molecule, and so that's introducing some biases and another chance to make false predictions. So I think there are some ways in which chemistry representation could be improved as well that would be helpful within the sphere of 3D representations to give the methods better substrate on which to work.
Awesome. Well, Paul, I really appreciate the time. This has been a great conversation. I've really enjoyed getting to get the benefit of your expertise, and I look forward to connecting down the line.
It's been a real pleasure. Thanks very much for inviting me on the podcast.
Paul, thank you. This was a real pleasure. What I'm taking away from this is more interesting than the usual story about AI and drug discovery. That story tends to be a fight. Old methods versus new ones, classical chemistry versus deep learning. Paul's whole career cuts against that. The methods chemists developed over the last 4 decades still work, and the best results come from using them alongside the new ones, not instead of them. 3 things stuck with me. First, synthesizability, which is easy to wave away and hard in practice. A generative model can hand you a great theoretical molecule, and changing a single atom can take it from something a chemist makes before lunch to something nobody can make at all. Paul's answer is to build the constraint in from the start. Start from the reactions that work and the building blocks you can buy, snap them together like Legos, and search that space. The obvious worry is that you give up novelty, but that space is so large that 99.999% of it has never been made by anyone, ever. The constraint barely costs you anything real. A lot of differentiation in drug discovery comes from the order in which synthesizability is brought into the picture, and it's interesting to hear how Paul and his team think about synthesizability as sort of a first-class constraint. Second, a lot of these models learn from a 2-dimensional drawing of a molecule. The thing that actually does the work in your body is a flexible 3-dimensional object moving around in water. So the model learns from an abstraction of an abstraction, 2 steps removed from the physics that matter. A lot of the work is figuring out how to make the model computationally efficient while capturing sufficient information to guide the selection and the assay cascade process that Paul mentioned. Third, a complicated model with enough parameters can look extraordinary, but end up measuring nothing but an artifact in your data. So the discipline that protects you is the boring part, which is why nobody does it. You have to benchmark the fancy method against a simple one, and you also have to do the grunt work of searching through your training, your validation, and your test set to make sure that there aren't any artifacts in your test set that your model is going to learn, or any data leakage that your model is going to discover. Paul and his co-authors demonstrated with the Hallison result that for the right problem, simple methods can do just as well as the complex methods, if not better, because they're not as prone to overfitting. And so that's the kind of case where methods decades older than deep learning can, recover the same answer or get you to a better answer. You can find Paul on LinkedIn. To learn more about Oxford Drug Design's work in antimicrobial resistance and oncology, visit oxforddrugdesign.com. If you know a computational chemist, a data scientist in drug discovery, or a team being pitched AI schools who'd benefit from a more rigorous lens on what they deliver, send them this episode. And if you haven't already, please subscribe to Data and Biotech wherever you listen. Thanks for tuning in.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.